Okay, so today we are going to finish digestion. And all right, so last time where we left off, we had been talking about up through the stomach. We learned about the functions of the digestive system. We had learned about the histology, the different layers of the digestive tract. We also learned that there's two parts of the digestive system. There's the actual tract, which is the tube from the oral cavity through the esophagus, stomach, intestine. We also learned about the accessory glands, right? The things that are technically not part of the tract, but they squirt material into the tract to help with digestion. And that would be things like salivary glands, liver, gallbladder, and pancreas. All right. So like I said, we spoke about the different organs, or at least we started off. We spoke about the oral cavity. What's in each organ has some features of it that enable it to carry out specific digestive functions. We learned about the oral cavity, things like the tongue and the teeth, which are important for mechanical breakdown. We learned about saliva, which has an enzyme such as amylase, which helps to break down starch. Then we spoke about the process of swallowing, deglutition into the stomach. We spoke about some pathologies, right? GERD, things like that related to that. If it's not working properly. Then we spoke a lot about the stomach, the anatomy of the stomach, the histology of the stomach, which is the histology itself. Remember, we've got those four layers, the mucosa, submucosa, and so on. Remember, the stomach is the one organ where there's three layers in the muscularis externa as opposed to two for the other organs. And of course, the stomach, the epithelium is organized into these gastric pits and gastric glands. And we spoke about the functions. Let's continue today with the small intestine. And even though every single organ is important, it's really crucial to say that the small intestine, this is where the bulk of digestion takes place. So even though starch digestion or carbohydrate digestion begins in the oral cavity and protein digestion begins in the stomach, the digestion of those nutrients is not completed until you reach the small intestine. So there's more breakdown of those in the small intestine, as is true with the breakdown of fats, right? So essentially digestion continues and is actually completed in the small intestine. This is also the organ where absorption begins. So all those nutrients that were too big to get into the blood, that were not sufficiently broken down, now they're small enough that they can actually be absorbed from the gut, the small intestine, into the blood. That would be things like carbs, proteins, or amino acids, and fats. The small intestine is about 8 to 10 feet long, highly convoluted, like even with the stomach, with the rugae for increased surface area. The small intestine, region-wise, there are three different parts of the small intestine. The first part, which is connected to the stomach, is the duodenum. This is where most of the absorption, most of the remaining digestion occurs. And then we have the jejunum, followed by the ileum. And the ileum is what will continue on into the large intestine. Excuse me. So we've spoken a lot about surface area, right, with the other organ systems, but also with the stomach. Remember, I mentioned that the stomach, we have the rugae, which is highly folded, which helps to allow for expansion, distension, also important for maximizing digestion. Same thing is true with the small intestine, where we actually have four distinct structural features that increase the surface area several fold. And we'll take a look at these. First of all, it's what we call the plicae circulares. Plicae circulares literally means circular pleats or circular folds. And this is what you would see in the small intestine. Kind of looks like rugae, but they're a little different in structure, and they're in the small intestine. So you'll only find those in the small intestine. Once you kind of get into the microscopic level, when you look into the small intestine, you'll notice that there's these protrusions into the digestive tract, into the lumen. These protrusions are known as villi. I like to think of them as like finger-like projections that go into the lumen. In between each villi, we actually have these invaginations called crypts. Crypts of Lieberkuhn. Some books call them intestinal crypts. These are very, very similar to what we call gastric and gastric glands in the stomach, but they're functionally different. All right, so we've got these plicae circulares, these folds. We've got these villi and these crypts, which again, more folding, more surface area. But then we have what are known as microvilli. And microvilli are located on the surface of the villi. So if we go back for a second, if these are the villi with these cells lining them, on the surface of each of the cells of the villi, we have these folds of the plasma membrane that are called microvilli. All right, so with all these 4 levels of folding, the plicae, which is really folding of the submucosa, with the villi, which is the folding of the mucosa, as well as we have the crypts, along with the microvilli, we have a massive increase in surface area, which is crucial if we need to absorb things, and that's what we do there, right? The greater surface area, greater absorption. That's why people who have a condition, which we'll talk about, known as celiac disease. Celiac disease is a reaction to gluten, as many of you guys know, very severe reaction, which results in the destruction of some of those microvilli, which impairs absorption. So you can actually become malnourished. All right, let's take a closer look at the plicae, villi and microvilli. Like I mentioned, the importance of all of these is that they greatly increase the surface area, important for nutrient absorption. The cell type we find here, which is really the same as the cell we find in the stomach pits, are simple columnar. And you might hopefully remember from anatomy that simple columnar is important to maximize secretion and absorption. All right, so I mentioned that the crypts are similar in appearance to the gastric pits of the stomach, in the sense that they're invaginating and they also have several different cells. But the cells, there is some difference. One cell type are mucus cells or goblet cells, where they produce a very alkaline mucus. And they function in protecting the intestinal tract from the acidic effects of food coming from the stomach. So much like the functions of mucus in the stomach, this serves like a protective role and also raises the pH. The raising of the pH is critical because the enzymes that work in the stomach work optimally when the pH is more alkaline. Remember, pepsin works best in an acidic pH of about 2. The enzymes that work in the intestine work best at around a pH of 8 or 9. So they need that alkaline environment. Other cells. We have an antibacterial cell called a Paneth cell, which are these ones at the bottom of the crypt. These produce enzymes that help to kill bacteria. So you can see there's a pattern here. We have saliva in the oral cavity that helps to kill bacteria, antibacterial enzymes. We've got a lot of lymphoid tissue. Remember I mentioned as we go along, clusters of lymphocytes, and we've got these antibacterial cells. So there's a lot of immunological effects or immunological mechanisms that are taking place in the GI tract because it is open to the outside world. The intestine is much like the stomach, but more so in the intestine. There's a high turnover of cells. When I mean turnover, a lot of replacement, a lot of renewal. Cells die very rapidly. The lifespan of cells in the intestine, as well as cells in the stomach, is very short. So we need to replace them. And so there's a lot of stem cells here. Fourth cell type is what we call enterochromaffin cells. These are basically similar to the G cells of the stomach in the sense that these make hormones. Remember the hormones of the stomach. We had ghrelin, we had gastrin, we have histamine, and we have somatostatin. Notice we have another library of hormones in the intestine, and we'll talk a little bit about what they do to help digestion. All right. So those 5 hormones, the ones mentioned on the previous page, let's start first of all with what's called GIP and GLP-1. Their function is to promote glucose uptake into tissues. So once nutrients go from the, remember I mentioned the intestine is where nutrients are absorbed from into the blood. Once those nutrients are absorbed into the blood, we need those nutrients to be uptaken into tissues, right? All throughout the body for energy, growth, and repair. These 2 hormones help with that, right? They help in the, essentially after a meal, helping glucose uptake into the tissues. And as we'll see, the effects are basically similar, very similar to the role of insulin, where insulin helps to move glucose into the tissue. Because you can have the best meal in the world, very healthy, you might absorb it into the blood, but if you don't have a way to get it into the tissues, you're going to starve and you're going to be malnourished. So that's the function of these 2 hormones. If you're wondering what they stand for, this stands for glucagon-like peptide. And GIP, oh God, I can't remember. Anyway, next 2 hormones were CCK and secretin. And they're important for stimulating gallbladder and pancreatic function. And we'll take a look specifically as far as what they're doing. All right. So let me just, I'm going to stop the share for a second and to just spell out the names of those hormones. So under whiteboard, all right. So we've got CCK. CCK stands for cholecystokinin. And so everything else I think is the same. GLP, P stands for glucagon-like. Why can't I remember GIP? It's something inhibitory, gastric inhibitory. I don't remember. You could probably find it online. I just can't remember. I'm a boomer. I forget things, as my son often says. Okay, stop share. And let's share again. Okay. Last one is serotonin. And serotonin, the last place you would think you would find it would be in the digestive tract, right? We already know it plays a very important in the brain, right? Mood, appetite, right? Kind of well-being, the target of many psychotropic drugs. We also know that it's important in blood clotting. Remember, it causes like the vasospasm or the vasoconstriction. In the gut, it actually stimulates the muscularis externa to contract to increase motility. So this possibly explains why some of those antidepressants, why one of the side effects can be gastrointestinal disturbance or disruption. All right. All right. Speaking of motility, I just wanted to show you a kind of a picture with what we mean with the different types of motility. And this is also true in the stomach. Peristalsis is essentially this wave-like propulsion of this contractions occur in a wave-like fashion that helps to push food downstream. Segmentation is the churning or the mixing that is, like I mentioned, tossing salad, or how a washing machine works, those types of things. So those two different actions can actually be disrupted by a lot of these psychotropic drugs because they interfere with serotonin. All right, so we've gotten through as far as the GI tract through the small intestine. And that's where I want to pause for a second and go into the accessory glands, because this is where a few of them act. This is a picture showing the duodenum, which is the first part of the GI, of the intestine. The stomach would be somewhere right up here. You can see that essentially 3 organs, the pancreas, this pear-like shaped gallbladder, as well as the liver, which you can't see up here, but you can see these ducts, secrete substances that travel right into the small intestine at the duodenum. The pancreas secretes a couple different things. It secretes a lot of enzymes, and when I mean a lot, per unit weight, the pancreas produces and secretes more enzymes than any other organ in the body. It secretes enzymes that help to break down proteins, carbohydrates, fats, DNA, everything. So whatever was started in the oral cavity and the stomach, the pancreas kind of helps to finish it as it secretes substances into the intestine. And of course, that's where the food is. It also secretes bicarbonate, which along with the mucus in the small intestine helps to not only neutralize the acidic chyme, so the food is pretty acidified coming from the stomach, but it also raises the pH because the enzymes that are coming from the pancreas work best at a high pH. So really 2 functions that are important to raise the pH. One is to neutralize the acidic contents of the food, which could damage the intestine if it stays acidic, and to raise the pH for, also raise the pH for effects of the pancreatic enzymes. What about the liver and the gallbladder? All right, well, liver has many, many functions, but from a digestive standpoint, its main function is to produce bile. And bile is an emulsifier that assists the fat digesting enzymes of the pancreas in breaking down fat. And we'll get into that a little bit later. So the liver produces bile, which travels down through these series of ducts, these hepatic ducts and the common bile duct. And I should back up a second. The pancreatic juice, per se, travels down the pancreatic duct into the intestine, into the small intestine. Bile is produced by the liver, but it's stored in the gallbladder and it's concentrated. So what happens is bile produced by the liver travels down this duct, but if you're not actively eating a meal, it doesn't get into the intestine. It basically backs up into the gallbladder where it's stored and concentrated until we need it, and then it's squirted out to digest fats. And we'll get into that whole process shortly. All right, so one of the things I mentioned, of course, is that the liver produces bile, right? But the liver also plays a very important role because the nutrients, remember I mentioned nutrients that are broken down, digestion is completed in the small intestine. After they're all broken down, they're absorbed. You know, thanks to the villi and the microvilli, they're absorbed into the blood, right? Well, when they're absorbed to the blood, where do they go? basically the first place they go is to the liver. So whatever you ate for breakfast this morning, or if you've had lunch yet, whatever you've eaten and it's been absorbed from the small intestine, it enters into the blood and goes right to the liver first, where it's processed before it enters into the general circulation. This is by way of a very important vessel called the hepatic portal vein, and I'll show you that in a second. Liver has many functions also. It also is a very important in detoxification of medication you're taking. So what do we know about the liver? It produces bile. It's the site where nutrients that are first absorbed from the intestine are stored and processed before they're released into the general circulation, and it also detoxifies. All right, so let's talk about this hepatic portal system, hepatic portal vein. All right, so what we call the hepatic portal system is a vasculature, and I'll show you on the next slide, that essentially delivers absorbed nutrients to the liver before they enter the general circulation. It's kind of like a weigh station. So what happens is the nutrients reach the liver, they're either stored there or they enter into the circulation. And this actually is also an important weigh station because, as we'll see later in this lecture, signals are sent from the liver to the brain that tells the brain what kind of the inventory in the liver. And if there's a lot of inventory in the liver, a lot of proteins, carbs, and fats, the signal to the brain is stop eating, or basically I'm full. If the inventory is low, the brain's gonna say, oh, I gotta keep eating. So it's kind of a neat little status report, which we'll get to later as well. All right, so this hepatic portal system, how does it work? Directly from the small intestine, we have a series of veins called the mesenteric veins. And these absorb nutrients from the intestine, those nutrients that were broken down there, thanks to the liver, gallbladder, and pancreas. From the from the mesenteric veins, they travel into the hepatic portal vein, which carries it to the liver. Now, what's interesting, of course, is, and this is kind of a fascinating thing. Normally, when we think of blood vessels going to an organ, we think of arteries, right? We think of arteries such as, the aorta and all of its branches. They go to specific organs to carry oxygen-rich blood. This is a blood vessel that's going to the liver, but it's carrying a lot of nutrients, but it's poor in oxygen. And I'll show you on a different diagram as well. Once this blood from the intestines that's oxygen poor, nutrient rich, reaches the liver, it's processed, then it enters into the general circulation via the hepatic vein, going to the heart and all the other organs. So here's a question. So all organs need a fresh supply of oxygen, right? Well, the only blood supply I've mentioned for the liver so far is the hepatic portal vein. But that's oxygen poor. Fortunately, there's another artery that branches off the aorta that's bringing oxygen-rich blood. So basically, you could say that the liver has a dual blood supply. The conventional one, which is the hepatic artery, which brings oxygen-rich blood, just like typical arteries do to the organ. And then there's the hepatic portal vein, which is bringing digested nutrients from the intestine to the liver where it's processed. All right. This is showing the hepatic portal system. Here we're talking about the small intestine, mesenteric veins. There is also absorption from the large intestine, but we'll get to that a little bit later. Most of the absorption from the small intestine is nutrients with a little bit of water and ions, whereas most of the absorption from the large intestine is really ions and water, no nutrients, no carbs, proteins, or fats are absorbed from here, from the large intestine. They're all absorbed from the small intestine. They are carried via the mesenteric veins. converge into the hepatic portal vein, which brings them to the liver, where the nutrients are either stored there for later needs, or they're released back into the hepatic vein, which goes to the vena cava, back to the heart. The aorta carries the oxygen-rich blood. All right. So that takes us to the end of the small intestine, but I did want to mention a little bit about a very important illness that can occur. Hold on a second. Oh, never mind. Let's see. Let me go back for a second. Oops. Just messed something up here. Oh, shoot. Hold on a second. Hold on 1 second here. Okay. All right. One quick second here. I'm just answering a chat. So, all right. Okay. All right. So let's back up. What did I do here? There you go. Okay, so we're finished with the small intestine. I wanted to mention celiac disease, which is one of many diseases of the small intestine, but it is one I think a lot of people are aware of. And just because of the effects of gluten, and gluten is very, the sensitivity to gluten, and we hear a lot about gluten-free restaurants, stuff like that. So what do we know about celiac disease? Well, first of all, before I get into celiac disease, I did want to mention that there are different types of gluten sensitivities. There's people can have a gluten allergy, which is different. People can have a gluten sensitivity, mild sensitivity, which is also different. Celiac disease is a full-blown immune reaction that creates havoc in the gut. And there's three conditions that have to be present for celiac disease to occur. All right, hold on just 1 second. I just want to, I have some little logistical stuff here. Okay. Okay, and then, sorry about this. Okay. All right. So 3 different causes for 3 different triggers that have to be present. First of all, obviously the offending particle is gluten. And gluten comes from a variety of different grains, primarily wheat, but also barley and rye, which have similar compounds, not exactly gluten, but they can cause a similar reaction. This is a protein that's present in the grain. Second of all, a genetic predisposition. Certain people, individuals, are just acutely sensitive to gluten. Not everybody is. So your immune system is already sort of primed to respond. Now that's 2 conditions, but really the final problem here is having what we call a leaky small intestine or a leaky gut. And I know that's kind of a nasty sounding term, but it is a condition. Normally the intestinal epithelium, this would be the cells, these are like the microvilli. They have these tight junctions that keep them closely bound to one another. Well, individuals that have a disruption of these proteins that allow kind of a free passage of gluten into the underlying tissue. Okay, hold on, one more thing. I'm sorry, I just say, okay, here we go. Okay. All right, so you got to have three things. You got to have the gluten, you got to have the predisposition, and you have to have a leaky gut. So what actually happens with celiac disease? All right, so a lot on this slide. I'm not going to expect you to know all the fine details, but we'll kind of navigate through. This is meant to be the intestinal cells. This would be like a villi, right? These are the cells, these are the microvilli. So you just ate some wheat or ate something, right? A grain. And with digestion, the gluten particle is released and it somehow gains entry due to the breakdown of these tight junctions, which are normally there, right? They're gone. The gluten gains entry underneath the epithelium. Once it gains entry underneath the epithelium, we have this connective tissue, you know, things like the lamina propria, the submucosa, where we have blood vessels, where we have lymphoid nodules, and those cells see the gluten right here and they go nuts. They basically explode. They go, what the heck are you doing here? Suddenly these white blood cells start squirting out chemicals to cause other white cells to come, because basically they see that gluten as a pathogen. So you've got all these lymphocytes, all these macrophages that set the whole process on fire. And ultimately what happens, as is true with many immune reactions, not only is the offending particle, in this case gluten, destroyed, but in the process, the intestinal cells are damaged. And you can see here. So when you destroy the intestinal cells, what actually happens is all that surface area for absorption is lost. So if someone does not find a way to overcome this, modify their diet or whatever, they could become malnourished because they might be eating a great meal, but those nutrients are not getting absorbed. All right. Moving to the large intestine. Large intestine, much like the small intestine, has several regions. Starts off with the cecum, followed by the colon, and then the rectum. Now, I mentioned before that the small intestine ended at the ileum, and that leads into the large intestine. But much like what we saw with the esophageal sphincter, much like what we see with the pyloric sphincter, which regulates the flow of food from the stomach into the duodenum, the pyloric sphincter, we also have another sphincter that regulates the flow of material between the ileum and the cecum. It's called the ileocecal sphincter. Every region of the digestive tract has its specific characteristics, as I've mentioned. I've mentioned, for example, that the stomach has 3 bands of muscularis, right? We've got the gastric pits. Small intestine, we're back to 2 layers of muscle, but we've got the crypts, right? And we've got all these different features. What is it about the large intestine? Well, first of all, in the large intestine, there are no villi. If we had villi, they'd be out like this. This is just the surface of the epithelium. So we have no villi. We have some of this you can't see in this diagram. I'll show you in a little bit. We have a lot of lymphoid nodules, which are more clusters of lymphocytes that help to kill bacteria as well. We have a lot of goblet cells. And I'm not sure if you can tell right here. Hopefully you can. You can see a lot of these, looks like holes or clear areas. These are all goblet cells. We still have the crypts, but instead of having all those specialized cells, they're primarily filled with goblet cells. What do goblet cells? Goblet cells produce mucus. Well, if you think about it, what would be a reason for mucus here, right? With the small intestine, the purpose of the mucus was to create an alkaline environment to neutralize the stomach acid that's coming in, and also to raise the pH so the enzymes will work. Well, in the large intestine, we're no longer dealing with absorption of nutrients. We're not dealing with all those enzymes. What we have now is a lot of, as we'll see later, a lot of absorption of water that occurs here instead of nutrients. So with the absorption of water, the waste, what's left, is becoming solidified. Without having the production of mucus, it would be very difficult for the waste to make its way out. So it basically functions to lubricate the lining of the large intestine. So the function of the mucus cells is to lubricate the lining of the large intestine. We have a couple other structures, which I'll show you in a second, called the Tinea coli. These are bands, an extra layer of muscle, which helps to push, move the solid waste. And we have these pouches called haustra, which I'll show. So these are all characteristics of the large intestine. All right, so what's occurring in the large intestine? Much like I mentioned before, this is where most of the water that's going to return to the circulation and the tissues is absorbed. So water, electrolytes, things like ions, right? Sodium, potassium, chloride, stuff like that, and vitamins are mostly absorbed here. There's no absorption of carbs, proteins, or fats taking place here. With the absorption of a lot of water, the waste is now concentrated, solidified, which explains why we need the mucus. And of course, it's solidified and stored until defecation occurs. All right. So I mentioned, of course, that we have regions of the large intestine. We have what's called the cecum here. We have what's called the colon here. And then we have what's known as the rectum. Well, within the colon, we also have different regions. The initial part of the colon, which is just adjacent to the cecum, is known as the ascending colon, because that's the way waste moves. Then we transition into the transverse colon, then into the descending colon, and then into the sigmoid colon. If you've ever had a sigmoidoscopy, this is where the scope, this is the extent of where the scope goes. If you have a colonoscopy, it goes throughout much of the colon. All right, so I mentioned the colon, or the large intestine in general, absorbs nutrients, well, absorbs water, ions, and vitamins, right? Then, of course, what's left is the solid waste, which is emptied into the rectum and eventually eliminated or excreted. Now, what's interesting here, and I want to pause for a second, we have a couple sets of sphincters, and unfortunately on this diagram, it only shows one. We have what's called the internal anal sphincter, which is about right up here. And we have the external anal sphincter. So we have two sets of sphincters. The internal anal sphincter consists of smooth muscle, much like most of the digestive tract, right? Except for like the esophagus and the oral cavity. The external anal sphincter is skeletal muscle. These two different sphincters must, normally they're in a contracted state. They must relax in order for defecation to occur, right? The internal sphincter, being that it's smooth muscle, is involuntary. So it's affected by just the pressure of the feces, which cause it to relax. The relaxation of the external sphincter depends upon our conscious will. So say, for example, you're driving along and then there's a rest stop, right? Suddenly you've got the urge to go, right? Well, the urge to go, it would be the internal sphincter. With the actual process of when you make that decision to defecate, that's the external sphincter, that's voluntary. So that's literally the end, no pun intended, of the GI tract. I wanted to expand a little bit more on the accessory glands and talk a little bit more about their role. As we already know, kind of in general, there's 4 types of glands, right? The salivary glands secrete saliva, which of course from a digestive standpoint contains amylase, right? We know that the liver secretes bile, which helps in the digestion of fat, and that the gallbladder stores and concentrates that bile. The pancreas secretes pancreatic juice, which, as I mentioned, contains a tremendous amount of enzymes as well as bicarbonate. All right, so let's take a look at the pancreas. First of all, the pancreas has exocrine and endocrine portions to it. The endocrine portion I'm not going to talk about. That will come when we start talking about the endocrine system. That was where insulin comes into play. Most of the pancreas consists of exocrine glands that are secreting the pancreatic juice. And like I mentioned, relative to weight, the pancreas produces more protein enzymes than any other tissue in the body. This is a list of all the different enzymes the pancreas produces. If we were to kind of compartmentalize enzymes that break down starches, which would be amylase, so it's kind of a continuation of what happened in the oral cavity. Enzymes that break down fats or lipases, enzymes that break down proteins or proteases, and then we have enzymes that break down DNA or RNA. Those are nucleases. So a lot of enzymes. And of course, bicarbonate is produced because that helps to buffer the environment, which helps to activate these enzymes. All right. So I mentioned those hormones in the intestine. Remember, I mentioned we have GIP, GLP, serotonin, secretin, and CCK, right? Well, the first 3, we already spoke about their effects on motility for serotonin, on uptake of glucose into the tissue. That would be GIP and GLP. But what about secretin and CCK? All right, well, these hormones target the pancreas. So how does it work? First of all, we don't want enzymes being produced at any time, right? Much like we found with the stomach, pepsin is only produced when there's food in the stomach, and then HCl gets secreted as well, right? That's the only time we want it. Same thing with amylase. So how does this work here? Well, when, for example, food is left the stomach and it comes into the intestine, as you know, from back, so the stomach would be up here, comes into the intestine. The pH, because of the hydrochloric acid, is very low, right? Right. And I mentioned, of course, that's where the mucus production in the intestine works. But what happens is secretin is stimulated. So when food comes into the small intestine, it's acidified, triggers the production of secretin from those enterochromaffin cells. That secretin travels to the pancreas by the blood and stimulates the pancreas to produce bicarbonate. And the bicarbonate helps to raise the pH. So very, very targeted effects. So those 2 hormones, secretin directly stimulates the pancreas to release bicarbonate. And then there's CCK. When the food that's entering the stomach, it's acidified, yes, but of course there are certain nutrients in there. And the amount of fat and protein that's in the food that's coming into the intestine is the trigger to release CCK. CCK travels to the pancreas and stimulates the pancreas to produce those enzymes. So if we go back here, remember, of course, we have the pancreas that produces bicarbonate under the influence of secretin from the small intestine. So when the conditions are acidic, bicarbonate is stimulated by secretin. Those nutrients that enter the gut, that stimulates CCK to go to the pancreas and squirt out CCK. All right. A little bit more about the liver. We're obviously focusing on these latter two, right? The fact that it secretes bile, which helps to emulsify fats. And also nutrients are processed here, right? All those nutrients that entered through the hepatic portal vein enter into the liver and they're processed. But plus the liver, of course, has a few other functions. Recycling hemoglobin, as you guys know from our earlier discussion, also produces hormones and, of course, detoxifies. All right, so we know what the pancreas does. We also know what the liver and the gallbladder do, but let's take a look. Let's kind of connect the liver and the gallbladder somewhat. So liver makes the bile. It synthesizes the bile. And of course, the bile is a byproduct of that bilirubin that comes from the recycled hemoglobin. Remember? And also there's cholesterol in it. I apologize not having that in here, but think of bilirubin and cholesterol. And the reason why I'm saying that this is going to be significant as we go along. The gallbladder stores the bile, okay? The bile that's traveling from the liver and traveling from the gallbladder basically travels through what's called the common bile duct. And from the common bile duct, it travels to the duodenum. So if we go back here for a second, here's the common bile duct, right? So all the pancreatic stuff goes here. All the stuff from the liver and the gallbladder travels through that common bile duct. Oops. All right. The common bile duct joins with the pancreatic duct to form a structure called the ampulla of Vater, and you'll see that on the next slide. At the ampulla of Vater, there's another structure called the sphincter of Oddi. Reminds me of the dog in Garfield, but it's spelled differently. And the sphincter of Oddi, much like any other sphincter, when it's open, it allows substances, the pancreatic juice from the pancreas, as well as the gallbladder, to flow into the duodenum. And this typically occurs when there's food in the intestine. When there's no food in the intestine, the sphincter of Oddi is closed. So let's take a look at this. So kind of putting it all together. Here's the pancreas. Here is the pancreatic duct, right? That squirts out either bicarbonate or enzymes or both once food enters the small intestine, right? From the small intestine, the hormones enter into the blood, stimulate the pancreas to secrete either bicarbonate due to the low pH, as well as enzymes due to the fat and protein content. And it goes down this pancreatic duct, okay? Bile from the liver passes through the common bile duct also, as well as which enters into this common pathway called the ampulla of Vater, this kind of a collection point. And then we have the sphincter of Oddi. So when you're not eating, this food or when you're, after you're done digesting, the sphincter of Oddi is closed. When food enters into the intestine, the sphincter of Oddi is open. And so we have entry of everything. All right. Now I want to talk a little bit about the bile, and I want to talk a little bit of how the gallbladder is regulated, because that's also under hormonal regulation. So there's a few different diagrams here. Hopefully it's not too confusing. One of the things I wanted to mention is, of course, high cholesterol, or specifically high levels of LDL, are a risk factor for heart disease, right? And even though some people watch their diet, limit their intake of dietary cholesterol, their cholesterol levels are still high. And that's because our liver normally makes cholesterol anyway, which is incorporated into some hormones, but also it's incorporated into bile. So one of the strategies, a couple different strategies for lowering cholesterol, is a drug called a bile acid sequestrant, which basically promotes the excretion of bile. Much of bile is normally reabsorbed, but with the bile acid sequestrants, instead of it being reabsorbed, it promotes it, more of it to be excreted. So when you excrete more bile, you're actually, as opposed to reabsorbing some of it, you're actually lowering cholesterol levels. There's also other drugs, such as you may have heard of statins, and statins are what we call HMG reductase inhibitors. HMG reductase is an enzyme that's the rate-limiting step to making cholesterol. So it basically acts on this enzyme, lowers cholesterol levels. So this is kind of really not part of this discussion in terms of what we're talking about now, but this is really more relevant. So one way you could lower cholesterol is by increasing the amount of bile that's secreted, because it contains cholesterol. All right. So last organ I want to talk about, and then we'll get into some other key interesting mechanisms, is the gallbladder. So we already know what the gallbladder does, right? We really know about what all organs do. But in addition, we also know in the case of the pancreas that it's regulated by CCK and secretin, right? When food comes into the small intestine, secretin is released, as well as CCK, and it affects the pancreatic secretions. Well, I did want to say that CCK also affects the gallbladder. That same CCK that's going to the pancreas also acts on the gallbladder, stimulates the gallbladder to contract. When the gallbladder, because it's a very muscular organ, when the gallbladder contracts, it squirts the bile that's stored and concentrated through this duct called the cystic duct into the common bile duct and then out. So I'm going to show you a video from today's standard. The graphics aren't that great, but I think it's pretty simple and informative. So let me see if I can get it to work here. Oh man, what the heck? I just got a new computer. What the heck is going on here? Let's see. I already do have Flash, I thought. Okay. How's going with Flash animations requires current Flash player. All right, well. All right. Well, I guess I got to update my Flash. Sorry about that. If I get it figured out, I'll go ahead and send it to you. It's a pretty cool video, but let me just get back to where I was here. Basically what it shows how when food, primarily fat, comes into the small intestine, it triggers CCK, which besides the effect on the pancreas, triggers the pancreas or the gallbladder to squirt out bile. And I'll have to check that out. So, all right. Now we're sort of on the final, well, so now here's one little thing just to summarize what happens with the gallbladder. It's a muscular organ attached to the inferior surface of the liver, stores and concentrates bile. Sphincter of Oddi is closed when the duodenum is empty. Bile that's released from the liver backs up into the gallbladder because that sphincter is closed. If your diet, and usually some aspect of our diet contains fat, enters into the small intestine, this triggers the release of CCK, which goes into the blood, stimulates contraction of the gallbladder because it's very muscular, which squirts bile into the common bile duct. And at the same time, CCK also promotes opening or triggers opening of the sphincter. So it's really doing three things. It stimulates the release of enzymes from the pancreas. It stimulates the release of bile from the gallbladder. And most importantly, of course, it opens up the sphincter, because all those secretions aren't going to do any good unless that sphincter is open. All right. All right. So just a little bit about liver anatomy, as long as we're talking about the liver. I did want to mention a couple different key things about the liver. And again, there's a lot of stuff on this slide. What I basically want you to know is that the cells in the liver that manufacture bile are called hepatocytes. Once bile is first made by the hepatocytes, it enters through a series of tubes called canaliculi, which eventually drain into the common bile duct. And you'll see that on, oops. So here it doesn't show the canaliculi, but the bile that's produced by the canaliculi goes to these series of ducts, ultimately draining into the common bile duct. And there's the whole ampulla and all that stuff. All right. So that's the main cell. The primary cell in the liver is the hepatocytes. I did want to mention one other cell you don't hear about much. It's called the Kupffer cell. And the Kupffer cell I would compare to the microglia of the brain or the alveolar macrophages of the respiratory tract in the alveoli. These are phagocytic cells that help to kind of remove any debris, kind of clean things up. So two cells. Most of the cells are hepatocytes that make the bile, and then the other cells are the Kupffer cells. This is also an interesting diagram because it shows the two blood supplies. Remember I mentioned that the liver has the hepatic artery, which supplies it with oxygen, branch off the aorta. It also contains the hepatic portal vein, which is delivering all those digested nutrients to the liver where they can be processed. And that's, of course, oxygen poor. This is just another diagram showing it all. All right, this is a summary of basically what we just went through, so you can kind of look at that a little bit, kind of as a summary of what all the different digestive organs are doing, as well as including the accessory glands. All right. So last two things I want to talk about are a couple very interesting physiological processes. One is understanding that there are three phases of digestion. So all these different things that I've been speaking about, the secretion of saliva, the production of pepsin, CCK, all that stuff, right? That is not always occurring, right? We want it to be very tightly regulated. We don't want all this stuff squirted into our gut at the same time. We only want it occurring when we're eating or when we're about to eat. All right, so having said that, there's three main phases of digestion. The first one is what we call the cephalic. And that, by the name itself, that means brain. So this is a phase of digestion that occurs basically when you're thinking about food, if you smell food, if you see a nice burger on the grill, whatever. We're all very familiar that when we think of food or whatever, that gets the saliva going, right? So not only does it get the saliva, but it gets the gastric juices, all the pancreatic. It gets the juices flowing, literally. Even though we don't have any food yet in the stomach, or even in the oral cavity, the body is getting ready. Because we're thinking about this involves the brain, the cerebrum, cerebral cortex, like the telencephalon, the hypothalamus, as well as the medulla oblongata, because the medulla oblongata is what's going where the vagus nerve fibers are going to originate from. The vagus nerve, as we've already learned, that's very large nerve that encompasses the entire digestive tract, as well as heart and lungs. So when all these senses are coming into play, that gets the medulla oblongata via the vagus nerve squirting out acetylcholine, and that's going to get things going, right? As food enters the stomach through the lower esophagus, so I kind of skipped the esophagus, but you can kind of picture this still occurring right here. As food enters the stomach through the lower esophageal sphincter, the stomach starts to sample things. And this is very interesting. We don't normally think of taste receptors in the stomach, but it's been determined that the stomach actually has taste receptors, much like we would think of as on the tongue, or chemoreceptors that know the chemicals. We have chemoreceptors and we have stretch receptors. What actually happens is the same parasympathetic pathway that started the process starts to stimulate more gastric motility and further increase. So basically what it's doing, it's sustaining it, and it's sustaining the vagus nerve activity, because what's happening is as the food enters the stomach is starting to stretch, right? Flatten out those rugae, and that in turn is activating more of the vagus nerve. So the trigger for the vagus nerve is different, right? Here, it's literally the sight, smell, and thoughts that are triggering it, and via the cerebral cortex. Here, it's literally through the presence of food in the stomach. That's kind of you want it to continue, right? You don't want those juices to stop flowing just because food enters the stomach. Lastly, the intestinal phase. I spelled chyme wrong. It should be C-H-Y-M-E. As food leaves the stomach, enters into the duodenum through the pyloric sphincter, we also have more chemoreceptors and stretch receptors, but these are starting to occur in the intestine. So food is now gradually leaving the stomach. So until it completely leaves the stomach, we're still having gastric secretions, but now the stomach is starting to empty. And as the stomach starts to empty, we're starting to see intestinal digestion occurring, again, through more pancreatic, hepatic, and gallbladder secretions. This is actually due to the same process, the vagus nerve. Now, what's happened, so we've got the gastric secretions continuing for a bit, and then until it gets into the intestine. But then we have an interesting thing that occurs. Those same hormones that we've spoken about earlier also inhibit gastric secretions. Why would we want them to inhibit gastric secretions? Because now that food's in the intestine, we want the stomach to shut down. We don't want hydrochloric acid to still be produced in the stomach when there's no food in there, right? Because that would create a problem. So looking at a visual of this process, this is basically what it's talking about. I mean, they go into a few other things. They mention histamine. Just go by what I mentioned on here. Oops, what the heck? On here. So go by this. But then you can see these three phases. So cephalic phase, again, mental input. Here we're looking at the gastric phase, which involves kind of the food in the stomach that's being stimulating more vagus nerve, more production. And here we're looking at the intestinal phase, where we're starting to see a little bit continuation of what's going on in the stomach, but then these three hormones begin to sort of inhibit the stomach. So it's pretty cool how that works, I think. All right, we're finally at the end here. Well, almost. And this is an area that's been very fascinating to me. I try to update my lectures because the story keeps not changing completely, but more components are added to it. You know, you often wonder what makes us feel hungry and what makes us feel full. We already had a little taste of that when we were talking about the endocrine glands of the stomach, endocrine organs, the production of ghrelin, right? We spoke about how ghrelin stimulates appetite. Well, before we get into all the details, I wanted to show you this picture and look at a couple things. Number one, note the direction of the arrows, right? For the most part, these arrows are going from a particular organ, a digestive organ, whether it's the pancreas, whether it's the stomach, whether it's the intestine, and they're going up to the brain, right? We all know about the gut-brain connection. The other thing to mention is notice the color. We have several things that are colored in red, one thing that is colored in green. This is significant. The green color indicates something that's stimulating appetite. The red indicates things that are inhibiting appetite or making us feel full. So if you look at this, you think, well, gosh, you know, we have a pretty good system in place that should prevent us from overeating. However, this does not take into account other factors, like says, you know, psychological factors, stress, right? That can kind of override this whole thing. But the basic storyline is in place. So let's take a look a little bit as far as what the process is involved. All right, first let's take a look at the induction of appetite. So as you already know, probably during the, probably prior to the cephalic phase, although sometime prior to when we're eating, ghrelin is produced by the endocrine cells. And that ghrelin enters into the circulation, because it's a hormone, and goes up to the hypothalamus and acts on what are called orexic neurons, not to be confused with anorexic, right? This is the opposite of anorexic. Anorexic means, right, not eating. Orexigenic means stimulating to eat. And these neurons stimulates the neurons in the hypothalamus to release a hormone called neuropeptide Y. So this is the induction of appetite. This is making us, and then once neuropeptide Y is released, that tells us, oh, it's time to eat. What about the other side of the coin? Mechanisms of satiety or satiation or feeling full, right? So as you saw from this previous slide, right, we have a lot of red there that tells us that we're full, right? Well, first of all, much like with ghrelin, we have things that are reported to the brain from the stomach and the intestine. When the stomach distends or stretches, or when the intestine distends or stretches, this sends a signal to the brain. And I'll explain how that works. So the stomach and the intestine have a role in making us feel full, because as more food is in these organs, the more the organ stretches, this basically tells the brain, gives an indication that I'm already eating, I don't need to eat anymore. In addition, the nutrient status of food in the liver. Remember I mentioned that the liver is sort of like it's where the inventory is. Food comes into the liver from the hepatic portal vein, and what happens is information from the liver goes to the brain finds out how much protein you had, how much carbs, how much fat. All right. So hormones are also involved. In addition to the liver and the stomach and the intestine, adipose tissue, which is, why is this doing this? Which is fatty tissue, produces a hormone called leptin, right? Leptin is a hormone that travels to the brain. Insulin is a hormone from the pancreas that also travels to the brain. PYY is a hormone from the intestine that travels to the brain. Now, where do they travel? Well, they travel to the same region, the arcuate nucleus, right? Like you saw back here. But instead of stimulating orexigenic neurons to produce neuropeptide Y, they stimulate what are called anorexigenic neurons to stimulate the release of what's called alpha MSH. And it's alpha MSH that promotes the sense of fullness. And to kind of cover their bases, not only do these three hormones, leptin from fat, insulin from the pancreas, and PYY from the intestine, not only do they stimulate alpha MSH to promote satiety, but then they go back to where neuropeptide was produced and basically shut that down. Pretty cool, I think. All right, so this is a nice little summary, and we'll kind of hopefully walk through it pretty easily. So this is the stomach, produces ghrelin. You notice it's ghrelin from the stomach goes all the way to the orexigenic neurons, stimulates the release of primarily what it says NPY. There's another substance, but it's primarily NPY. You notice it has a positive effect, right? Then I mentioned there's several hormones. There's leptin, which inhibits those same neurons. The pancreas, as well as PYY, not only inhibit the neuropeptide Y, but they also stimulate the production of alpha MSH, where the heck is alpha MSH? Actually, okay, alpha MSH is a byproduct of what's called POMC. Yeah, POMC is the precursor, but you could just say alpha MSH. The POMC is a large molecule that's broken down to alpha MSH. So these insulin stimulates this anorexigenic neurons, which stimulates the release of alpha MSH. Leptin does the same thing. So anyway, hopefully that clarifies it. If that didn't clarify it, this pretty much does the same thing. You notice the red, leptin, PYY, and insulin, negative, ghrelin, positive as far as appetite. Final slide I wanted to mention, this is interesting as well. Leptin was actually discovered through some genetic research where they were studying strains of mice. And these two mice are litter mates. In other words, they're either two brothers or two sisters or a brother and a sister. Same mother, same father, everything. You notice they're very different. And they were both, started off very much the same. But look at the difference. This guy is huge. This one's very little, right? This guy has a gene mutation that results in it not making leptin. So this guy doesn't make leptin. So what does that mean? Going back to here very briefly, by not making leptin, even though it's making the other stuff, it's not getting the signal to stop eating, right? It's not doing this. So as a result, it eats and eats and eats and eats. Normally what happens once you eat a certain amount, leptin is produced that tells you you're full. This guy's not getting that signal. So the way the body should work, this is meant to be like adipose tissue. As you eat more food, or as if you gain too much weight, there's a production in leptin, and the more leptin you make, normally that kind of inhibits the food intake. It's sort of like a self-regulating system. If you lose a lot of weight or don't eat much, this lowers leptin, which essentially triggers you to eat more. So normally we have this self-regulating system, but obviously there's other factors that come into play, like I mentioned, psychological, maybe certain illnesses that can affect people. So that takes us to the end of, oh, everyone's still here, yay. All right, so that takes us to the end of the digestive lecture. Anyone have any questions at this point before looks like everyone's pretty much on mute. That before I just go into the review. Any comments? I'm going to go under where it says if everyone has any comments. No comments. No comments. Let's see. All right. Okay, so I'm just going to go into the review. Hopefully I still have it here. Otherwise I'll have to, let's see. Yeah, I guess I got to go here. And wrong class. 235. There we go. Okay, let me go back to Zoomerama here. Okay, what the heck? Okay, and then new share, and then Everyone should be able to see that. Okay, this is bizarre. Hold on one second. Okay, stop share. Okay. All right, so hopefully everyone can see the review. And stuff should be pretty familiar to you as we're going through it. Basically, to start things off, we covered the digestive system. In the very beginning, for example, we spoke about what are the general functions. There is that first slide. Remember, digestion, motility, secretion, absorption, and then excretion. Let's see, why is I get that touch it every freaking time. There we go. Then we hold on 1 second here. There we go. All right. Types of motility. You should know the types of motility, speaking that, especially the fact that that's one of the functions of the digestive system. Motility, remember, that's primarily via the muscularis externa. Peristalsis is the propulsion of food through the GI tract. Segmentation is more of the mixing or tossing salad. We then spoke about the histology of the GI tract, so you should know the different layers, the sublayers, like for example, remember the mucosa contains the epithelium, lamina propria, all those. What is the important function of each layer? Also, of course, you should know what the regional changes are. So for example, remember, no matter what, there's the 4 layers, but remember in the esophagus and the oral cavity, remember the muscularis is skeletal muscle, and then we transition into smooth muscle. Remember in the stomach, there's 3 layers of smooth muscle in the externa, things like that. We have gastric pits, villi, whatever in the intestine. So just know the changes in the histology with the different regions. While we were going over the regions, we also spoke about the enteric nervous system and those different nerve plexuses. Remember the submucosal and the myenteric. Know about what they do. Remember that sometimes referred to as the second brain, meaning that it really can control the GI tract. However, it is influenced by the ANS, as you know, right? Parasympathetics can increase digestive function, sympathetic can decrease it, but without those 2 inputs, it would still function on its own, much like the heart, right, with the conduction system. All right, next we went through the organs of the GI tract, and basically I just put that, just know all the organs of the GI tract, organs of the GI tract, their structure, and their functions, the structure of the intestine, the structure of the small intestine, large intestine, stomach, right? All the way from the oral cavity all the way through the colon. Some of the modifications that I mentioned before with the histology, know about the gastric glands. What are the cells of the gastric glands and what is their function? Remember, one of the cells is an endocrine cell. So know that cell produces 4 different hormones. You should know the primary role of those hormones, whether they increase digestive function, whether they decrease digestive function, whether they promote appetite. One of the cell types is parietal cell. And of course, we know it produces hydrochloric acid as well as intrinsic factor. We went through a video as well as describing how hydrochloric acid is made, those different steps. couple different processes. One of the processes that we spoke about was the process of deglutition, and of course that occurs way back between the oral cavity and the esophagus. Remember that there are 3 phases. You should know what occurs in each phase, the buccal phase and so on. Once we swallow, of course, you know, we have those sphincters in the esophagus, the upper and lower. We spoke about a condition that is related to a defect in the lower sphincter. So you should know a bit about what GERD is. And of course, how we treat it, how it can be treated. Continuing with diseases or pathologies, we spoke about not only pathologies of the esophagus, such as GERD, but pathologies of the stomach, such as peptic ulcers. There are others. So, one of you was kind enough to send me some information, which I think I'll include next semester, about Hirschsprung's disease. But anyway, for now, peptic ulcers. You should know about peptic ulcers. What are the causes? Remember, one of them is H. pylori. And you know why H. pylori is able to thrive in the stomach, whereas most bacteria cannot. Now, the stomach, of course, is the site for protein digestion, and of course, we have to have an acidic environment, but to break down the proteins. But the problem is we have this very delicate epithelia that if you were to like pour out hydrochloric acid onto it, it would burn them up. So we did speak about different strategies we have, namely the production of mucus, the regulation of mucus production by prostaglandins, and how they protect the gastric mucosa from the effects of acid. Moving from the stomach into the intestine, we have the intestinal crypts, which again, comparable to the gastric pits in a way, but different cells. Know what those cells do, what hormones are made by those enterochromaffin cells, right? We spent a lot of time talking about the digestive tract, but obviously equal amount of time should be spent on the accessory organs. You know, what those organs are, what they make, the different types of salivary glands, how the pancreas and gallbladder and liver, their anatomy, their functions as far as, obviously the liver, a couple different functions, right? One is its role in the absorption of nutrients from the small intestine via its hepatic portal system, but also, of course, along with all these other organs, their role in the production of substances that enter into the duodenum via hormone stimulation. So their contributions to digestion. Like the endocrine control of those organs, features of the hepatic portal system, know the names of the vessels, where they go, where they start from, where they go to, are they oxygen rich, are they oxygen poor? Finally, celiac disease, which is not related to the accessory glands per se, but it's related to a very strong immune reaction to gluten and other wheat-based proteins. So we spoke about that, what happens, and what are the necessary ingredients to trigger that. Histology, histology, I guess, of the rectum, anus, and defecation. I spoke about, of course, how the rectum is where feces are stored. And of course, I did speak about the sphincters, which play a role in defecation. The external anal sphincter and the internal anal sphincter, know the differences, voluntary versus involuntary, right? Smooth muscle versus skeletal muscle. The last two parts, which should be well entrenched in your memory, of course, are the phases of digestion. And then finally, the endocrine control of appetite and satiety. I did want to do one last thing. So that takes us to the end of the review, but I did want to go back and draw something out for you because I realized that I left one thing out. and it's on my whiteboard. I spoke about the liver, how it plays a role, how bile is important in emulsifying fats, right? So what is emulsifying fats? Well, first of all, let me just sort of, all right, when I'm saying emulsifying fats, it's basically fat. If you take fat or oil, which is fat, add it to water, you'll notice that the fat kind of blebs, right, in these little droplets. So here's another droplet. And I'll draw another one. All right, so these are fat droplets. All right. All right. Now, in the presence of both two things, in the presence of both bile from the liver and also lipase from the pancreas. No, actually, let me back up for a second. Let's just do bile first. In the presence of just bile, right, those fat droplets become very small. Okay, so this is what I mean by emulsifying. It's still fat, but instead of having, and there's going to be more. I'll just draw more just to make it a bit more realistic. So those three lab fat droplets got broken down to small ones. That increases the surface area such that when lipase comes in, it completely breaks down the fats into what we call, I'll just write this, fatty acids, and glycerol, or essentially completely breaks them down. So we don't have any more droplets. Okay, so remember, bile, of course, is produced by the liver, but it's stored in the gallbladder. And then lipase comes from the pancreas. So it's kind of several different partners help out with this. So hopefully that helps out if you were wondering about that. All right. So that basically ends our lecture and review. Does anyone have any questions they want to ask? It looks like almost everybody's on mute here. Let's see if I can see anyone who's not on mute. Everyone's on mute. They must have really noisy houses, maybe barking dogs like I do, or noisy kids, or just listening to the radio, something. All right, so if no one has any questions, what I will say is the exam is next Tuesday. So I will have office hours on Monday. Let's say Monday at, hold on a second, let me check this here. Monday. I can do Monday at 10 o'clock and then Monday at 3. So I'll just send you guys an email about that. Monday at 10 and Monday at 3. So other than that, we're good. I assume everybody is happy or
I have
not
a question.
Sure. Oh, there you go. Yes.
I was wondering, are you going to pose some of those practice questions again? Or?
Yeah, I will send some out. Yeah, I will send out like a short little practice exam. Sure.
Thank you.
Sure. That was Carla, right?
Yes.
All right. Anyone else? All right. Well, I will see you guys Thursday. I will see you on next Tuesday or Monday if you have office hours. You can always email me if you have any other questions. All right. Have a good weekend.